The cytoplasm, viscous, clear stuff inside the cell containing organelles. What is an organelle? What's an example of an organelle?
An organ.
Huh?
The heart or the...
No, organelle, not organ. The Golgi apparatus.
Oh, okay.
What makes what an organelle? How do we define
A group of tissues linked together.
Actually, we're talking on a cellular level. We haven't gotten to our tissues
I mean, a
yet.
group of cells.
We're talking about one cell, right?
What's your question?
What is the definition of an organelle?
It's the material within the cell.
We got that part, but it's
the
the
cell.
What else? What
They
part?
don't have specific functions.
Yeah, they do. But the basic definition, to sum it all up, is they all have a membrane within the cell. Golgi apparatus has a membrane in the cell. Endoplasmic reticulum has a membrane in the cell. Mitochondria has a double membrane in the cell. Right? So any component within the cell that has a membrane around it inside of the cell is called an organelle. Prokaryotes can't have organelles because they're about the same size of an organelle. So that's distinguishing them from your eukaryotes. Eukaryotes are bigger than prokaryotes. So basically what you're going to find in the cell is plasma membrane, the cytoplasm containing its organelles, and the nucleus. And you guys told me already, cell metabolism, synthesis of molecules, cell communication, replication. So you pretty much have the functions of cells. It would probably be denoted as eukarya, and actually I'm speaking from your earlier biology class, but eukarya is E-U-K-A-R-Y-A. Yeah. And you can look it up. That's a term that if you're not familiar with it, you might have forgotten it. It's the term that's been discussed through all your biology classes. Your 121s discuss it. Your 100 levels discuss it. Because there's two cell types that we know to date. Prokaryotic cells and eukaryotic cells. And bacteria and archaea fit in the prokaryotic cells, and eukarya is the category that has the kingdom. Kingdom protista, kingdom plantae, kingdom animalia. Well, kingdom fungi, protista. Kingdom plantae, and kingdom animalia. That whole thing about the plasma membrane, it denotes what comes in and denotes what comes out. It is selective. So some things can come on through without any issue. Some things got to be transported through, and some things need energy to be transported through. So this deals with active and passive transport when we talk about the plasma membrane. The plasma membrane has this mosaic model made up of the phospholipids that I tried to draw earlier, tails facing each other, and the heads facing the water environments, which are polar environments. And integrated in there, you will find integrated proteins. You will find some carbohydrate chains connected to some of the proteins, and you will find some fats, some steroids connected. We pointed out that this is a phospholipid bilayer, and you remember that, and know that that's important for the membrane. And these are just some of the proteins that are integrated inside of, within the integument of the plasma membrane. There's some intrinsic proteins and extrinsic proteins. And they play a role of either transporting things in or out of that cell, or operating some mechanism within the cell by way of some chemical or some molecule connecting to the outside of the cell. And they have a whole category of this in your book. Let me get to that. Table 3.2, where it addresses the marker molecules, and you guys should have read or been reading over this. Attachment proteins, and you kind of know these. Transport proteins, which consists of the channels, the carriers, and the ATP power pumps, the receptor proteins, and the enzymes. These are all the different kinds of proteins that are integrated within the plasma membrane. And they all have different functions, as you but you can highlight this table to kind of get your boundaries down. So this whole thing about passive and passive and lab and getting no points for working together. Okay, so this whole thing with this DNA. So we got DNA. We've already said that it's got a double helix. And we're just going to put a 3 here. So let's say this is A, this is G, this is C, this is T, this is G and C. So what's the complementary relationship? This would be T, C, G,
A, C, G.
So always remember, bind together by what kind of bond?
Hydrogen bond.
Hydrogen bond.
A weak
Bonds are
bond.
easily broken. So this whole nature of the DNA. So then we say we want to replicate the DNA. We understand that replication happens in the S phase, the synthesis phase. That's where you get the DNA replication from. And at this point, all the DNAs are always going to be accompanied by an old and a new strand. This is what makes it semi-conservative. It replicates that way, incidentally. There's a few rules about DNA that you're always supposed to understand, and that's even when RNA is being replicated from, is that DNA replicates in a 5', 3' direction. That's how it replicates, 5', 3'. Even when RNA is picking up those nucleotides, it's picking up those nucleotides in a 5', 3' direction. All that stuff means is that on the 5th carbon, there is where you would have the phosphate. And on the 3rd carbon, that's where you would have the hydroxyl ion. I mean, it's chemistry, but you're to remember 5', 3' direction all the time. Some important things. There is one strand that is lagging, and there is one strand that's a leading. So you got a leading and a lagging strand. All the time. DNA replication occurs, lagging and leading. This one's no problem, because this one has no problem with a continuous 5', 3' direction. So this is continuous. No problem. This is what problem is. This is a non-continuous strand.